Well, I need my transparencies. Okay. Well, let me say that I'm not a physician. I'm not a biologist. I'm a physicist, and I work with optics, lasers, light, and all the effects related to the interaction with the atoms, molecules. Then we put a lot of molecules together. We make membranes. We put a lot of membranes together. We make cells. So we thought that we could do something there. But more in the applied part. So what we are doing is, we are just using light, and there are many techniques around the world to develop treatment of cancer, mainly, different types, and the microbiological control. And this is what I'm going to show to you as an example of things that physics can help in improving the application of cell biology to healthcare. So everything I'm going to say is in this transparency, basically. So we send light, and the idea is to provoke some reaction, either to kill the cell or to detect the light that comes back, and then we do diagnosis of disease. Okay, my group is I have a center. I coordinate a center with about 120 people, and more than half of those people are involved with biophotonics, which is light for life science. Okay, and everything I'm going to tell is in your homepage. So papers and everything, I'm not going to cite during the presentation, but you can find in your homepage. So we work with atomic physics. This is my main work in quantum fluids, quantum mechanics in the laboratory. And we have this biophotonics and nanoplasmonics. And the biophotonics is where we are concentrate in healthcare, point of care, home care devices, and new technology for that. And we care a lot about diffusion, innovation, and innovation with social responsibility. So in the biophotonics, we have a few laboratories, and basically we go from principles to instrumentation and clinical investigation. So we have 3 clinical bases, one in the cancer hospital in a city that's only cancer, and then we have another one in the humans hospital in São Paulo, and another one about mycosis treatment, onychomycosis, in another city. And basically, we are involved with the called photodynamic therapy. And if you don't know, photodynamic therapy is basically, I'm going to jump to this. It's a process that you put some molecules inside, you excite the molecule, there is an energy transfer, mainly to oxygen, generating a very reactive species, which is the singlet oxygen. And the singlet oxygen oxidizes almost all double bonds that are around and destroy membranes and taking the cell to death by necrosis. Okay. So for this to be well succeed, we needed to distribute light, because we not only treat superficial lesions, we sometimes treat bulk lesions, and life is not 2-dimensional, it's 3-dimensional. So we got to know very well light distribution and provide the correct light source for treatment. We normally need a molecule too, which is called the photosensitizer. It's the one that we excite to start the reaction. And we need oxygen. Oxygen nature gives us free, because you have abundant oxygen in many of the living objects. So what's missing for a success here, we can do very well light sources, and we can develop techniques to measure how light is distributed. But the photosensitizer molecule, we didn't have. And for this to be well succeed in a country like Brazil, we had to provoke companies to start to produce, and we have done. So now Brazil produces medications that are photoactivate medications, and it's being provided for many of the studies we do. Of course, my laboratory has a lot of students, so we do a lot of basic science, investigation of pharmacokinetics, how we destroy, how is the homogeneity of distribution of medication, especially in cancers. How can we predict the necrosis? How can we do the statistical distribution of cells that can be killed with one technique or not, and so on. But I'm not going to speak about this. I'm going to tell you about our clinical research. And basically, we are focusing in a few types of cancer for being treated with this technique. And again, we make molecules to go to the cancer cells, and then we illuminate, we provoke a photoreaction, and then the cancer cells die in minutes. This is a very fast technique. And we've been focusing head and neck cancer with a collection of doctors, intraoperatory hepatocarcinoma, and here's a pharynx type of cancer and esophagus, and skin cancer, because skin cancer is a big problem in Brazil. And we have more than 250,000 new cases of non-melanoma just in Brazil. I think Latin America as a whole is more than 500,000 cases a year. And this is a big problem because it involves many aspects, from economy to health to social problem. And there is a logistic problem. You know, Brazil is very big, and to treat the patients, we have to move them around. And this is a big cost. So if we have techniques that you can implement in different local and are easy to be implemented, this is a nice solution. And this is what we have done. We develop an equipment that's the first platform that involves fluorescence diagnosis for skin cancer together with the laser for treatment. And this is being developed together with a company that now produces the pharmacos. Now they have 3 pharmacos approved. And the skin cancer is the first one that we implement in Brazil. It's a very easy procedure. It's a single visit, fluorescence diagnosis, treatment, and this is a fluorescence of skin cancer, so you can see even if you are well trained, you see these red spots. You can identify quite well the size, make the dosimetry and everything if you do fluorescence-assisted treatment. The procedure is very easy. There is a kind of curettage, application of a medication, incubation of about 1 hour to 2 hours, and then the treatment. And the doctor, this is one of the techniques that the doctor, after the treatment, can know that if the necrosis is taking the volume or the domain that he predicts, or he can continue in the same section until he reaches the necrosis. So this is the necrotic, the same lesion that is necrotic, and it is a case that the doctor still have a red porphyrin around, and the doctor has to give continuous the treatment for being well succeed. Okay, so we have treated more than 6,000 patients of small lesions in Brazil, and this is now a technique that's being exported. We didn't invent it, this is being around, but I think we did the biggest multi-center and the demonstration that this can actually replace surgery if it is well on the hands of well-trained physicians. And I'm just jumping very fast to the cases. What are the results we have? So at the present, for patients that we are following for more than 36 months, we have more than 95% complete elimination of tumor with a single visit. We developed the protocol, the drug, optimization of the drug, and now we are implementing in many countries. This is Pakistan. And in Brazil, we have about 100 localities that our center is coordinating. And now this is already commercial available. And we have about another number like that of private clinics that are doing the treatment. So those are some of the centers. We made a national program that involved training the doctor and providing the medication and assistance for the first 20 patients that he treats. So we send a group of people there. And after that, he has to keep going and report the results to a center. So many centers are there. The medication, this is now being approved in Mexico by COFEPRIS. So we start many centers in Mexico, and all the countries I listed here are Latin America countries that we have now partnership. And we have even partnership with the United States. You know, even the United States, with a lot of money for healthcare, they are starting to think that treating skin cancer with photodynamic therapy may be good cost benefit for the insurance, so they are paying attention. We are in Venezuela, Peru, and all those countries, we are there. And I think this is an example of things we can do together for healthcare. Now, the cancer has been around for many times, the photodynamic, but the microbiological control is where this technique can make a big difference. And why can make a big difference? Because, you know, you have the superbugs, the superbacteria resistant to antibiotics, and this is a possibility of overcoming this problem. And we've been doing many researches, including blood decontamination for blood reservoirs. And you cannot see very well here, but all those diseases is being attacked with photodynamic. And the idea is very simple. We make sure a drug gets into the microorganism, and after illumination, we eliminate the infection. So this is a very nice, you see, you can decrease about 10 log orders in colonies of microorganisms, which is a very good indication that this is good. I'm not going to speak about this mathematical model that we have done for microorganism control. Everything is in a book we just published, collecting about 100 papers published and more clinical studies. And we've been attacking many problems. One of them is periodontic diseases. You know, periodontic diseases affects a lot of people, and biofilms. And with this technique, is just the placement in situ of the photosensitizer with illumination. We've been eliminating plaques and everything. And this is very nice because biofilms is very hard to deal with. But the oxidative effect of photodynamic therapy, I don't know if you can see, but it just removes the plaques. So this is a very nice thing that we are doing. Condyloma, HPV, is a national program. I think we are the only physicist involved in the humans' health care in Brazil. And we are treating condyloma in many places in Brazil. We want to do for the condyloma the same model that we did with the skin cancer. Companies that makes device, make the medication, and we go and treat the condyloma. And this is a program that's been well succeed. We made the device. The device is also being adopted now in the United States, in Maryland, in Johns Hopkins, for the treatment in Mexico. So we are partners there. And the result of this is being very good, you know, like the cervix lesion is divided in classes, degree 1, 2, and 3. Degree 1, they normally don't do anything in our country. And this is a mistake because when it's a recurrence of the lesion, it always recur in a degree above. So doing the photodynamic therapy in degree 1, cervix lesions, condyloma, this is prevent the evolution. So this is a preventive. And with this work, now we are doing the called NIC3 or SIN2 and 3 with very good results. We got many prizes, even Sanofi gave us a prize. The Mercosul gave us a prize. And there is another one I cannot read here. But we are now treating the degree 3 cervix lesions, which is considered carcinoma in situ, and the preliminary result is very good. Maybe we can prevent a young woman to lose their cervix, which is happening everywhere in the world. Now, we are also doing other treatments, like the ulcers, especially vascular and diabetics. This is a problem everywhere. You have always people with ulcers that cannot be healed in years. And we are doing a procedure that you decontaminate the ulcer and you add the artificial skin. And with that procedure, we're being able to convince the doctors that this procedure, in less than 2 months, they can recover ulcers that they could not do otherwise. So this is also a nice program. But here comes a very important one, which is the treatment of pneumonia with extra body illumination. We only did in rats so far, but the results is so great that we got money to do a first clinical trial. And the idea is very simple. We make the patient to breathe a photosensitizer by inhalation, and that goes to the colonies. In less than half an hour, we get all the colonies in the lungs impregnated with this substance, and by doing infrared illumination, extra body, we reach the colonies and we eliminate them. So in rats, you can see here the result. Groups of rats with pneumonia that we treat, no one die. And then the other ones that we try to do a regular treatment, they keep dying following those plots. Now we are doing for humans. And the idea is, again, we already have the drug that's easy to be carried by inhalation, vaporization and inhalation. And we have the machine to treat the first patients. So we hope we got the permission of the Ethics Commission in Brazil to start this procedure. And this will be very good because we have a lot of patients in intensive care units with developed pneumonia and the antibiotics are not working so well. So that could be a hope. This is another one that comes together with pneumonia. We find a way to prevent the formation of colonies of microorganisms in the tube of intubation. By having an optical fiber together, we keep the tube of intubation free of contamination. And that prevents the development of pneumonia during the intubation of patients in intensive care units. And this is it's so interesting that we have a company already that wants to buy our patent. And I think this is we functionalize the polymer that makes the tube, put a gel of photosensitizer and an optical fiber together, and there is no formation of colonies of microorganisms in that case. Well, the results are very good, and but here comes another one that's even better. You know, we have a problem of supply of blood, and by separating the components of blood, we can decontaminate each component and recompose the blood, the plasma, red cells, white cells, and we'll be showing here that we can get even hepatitis we can eliminate from the blood by doing photodynamic and photo treatment with UV in the plasma. Okay, so this is the idea of the thing. We separate and then we recombine the blood free of disease. This is a work we're doing with Toronto, which is decontaminating organs for transplantation. We're doing liver in Brazil and lungs in Toronto. And by circulating the liquid of preservation in the organ previously to put in the recipient, we can clean it up quite a lot. And this will increase the number of organs available for transplantation if we can prove and avoid complications by infection, which is the main complication in many cases of organ transplantation. Well, it seems very simple. The treatment of mycosis, you know, you have this called onychomycosis, and I didn't know about onychomycosis until I started to work with. And if I ask you all in this room to remove your shoes, we're going to find that 30% of the people here has a kind of mycosis on the nail. And turns out that people are taking fungicide. And the increase of hepatic cirrhosis because of use of fungicide is increases, including especially in human. Therefore, this technique that treats the fungus in situ is very good. It's only, you have a sequence of putting a urea type of cream and together, and after you make poros on the nail, and then you put a cream based on curcumin, a molecule that this company makes, and then you do the illumination, and look, in a few weeks you get rid of the fungus. And I think in the United States this is a big market because I know that Americans, they have fungus in almost all the nails of the feet. Okay, this is another one that we got a prize now, which is treatment of a throat infection in young kids, pharyngitis. And the idea is there is a kind of caramel, which is not a caramel, which has the photosensitizer, is made of hydrogel, and is in the process of being approved by Anvisa. And we give it to the kid, and in 3 minutes, this dissolve and impregnate the colonies of infection on the throat. And then there is a device that we developed that the doctor goes and illuminate. And I don't know if you can see, but in 94% of the kids that we treat, the disease disappears in less than 24 hours. As you know, there is a problem of abuse of antibiotics in treating infection, include young kids. Any young kid up to 12 months, they already try 2 antibiotics. And we know that the situation is getting very bad. And now we are fighting to go to the clinical, to the phase 3 clinical. We treat groups of patients in clinical 2. And we got an excellent result. And also for adults, there are adults with chronic throat infection. We know patients that are constantly taking antibiotic. And with this treatment, we've been very surprised that we can solve the problem in the majority of them. And now this is a very interesting case that we physicists try to convince the medical doctors that the kids will open the mouth for the light. And they claim that kids will never open their mouth. I say, they don't open their mouth for your instrument that looks like a knife. But a little light with going around, they're going to open the mouth. And 100% of the kids cooperate. They open the mouth for light. And the doctor goes and illuminate. We made a device together that the doctor can see by fluorescence the infection. Because by naked eye, you hardly can see. So fluorescence helps the doctor. And the treatment is so well succeeded that we got more than 10 phone calls a week of mothers that wants to do this treatment for the kids. But we are obeying the clinical trial and everything. But situation will be good. And Mexico, we presented this in a United States, in Boston meeting. And Mexico wants to start right away because they have a big problem there too. Well, Zika is a problem, but this is the problem, is the vector of the mosquito. This mosquito is smarter than people. It transmits 3 or 4 diseases, it's global, and make everybody pay attention to it. It's the dream of scientists. And how are we going to do this? Well, we are doing something that we develop a molecule that we throw in the little, because the mosquito procreates in water. So we are throwing a tiny little portion in the water, and then the mosquito absorbs. This is a confocal microscope of the larva, showing that we make all the digestive tract of the larva be impregnated with the photosensitizer, and we put this during the night. And when the sun comes, the light of the sun, even if it is not a direct illumination, kills the larva, and kills very efficiently by oxidation, and oxidize the rest of the molecules around, because the environment is normally very abundant in oxygen. So we eliminate the larva and we eliminate that molecule, so it is environment friendly. We are doing in the fields this, and we are well financed by the Zika program in Brazil, which I think he gave some example. And I think it's a program that in less than 18 months, Brazilian scientists came with answers, not to understand the disease, but also to propose techniques that will help in many cases. Okay, well, for my friend in Mexico, you may know this company in Mexico that's called La Cienta, is the largest exporter of broccolis in Mexico to the United States. And they have a problem of contamination because they reach the level of hyperacetic acid they use, and FDA say, you have to put this half. And for some reason they call us, we made a machine, and without using hyperacetic acid, we put the amount to 10%. So we somehow surplus another 10 years of requirement of FDA. And the idea is very simple. I'm not going to have time to explain to you. So we are acting now in the food industry because it is a very important industry in Brazil and everywhere in the world, especially in Latin America. We are exporter of food. And we are worried also about decontamination of environment, especially surgical environment. And the technique is so important that we want to introduce photomedicine to everyone. We made a kit, educational kit of high level for university level that anybody can get. I'm convincing Trieste here in Italy that we should make a course and give it to everyone that comes a kit like this, so they go home and start to do experiment with this. Well, you see, just to conclude, we are very worried about cancer because, you know, cancer was never treated so well. We have so many new techniques, so good ways of killing cancer, but it's very interesting that it was not so less available for people. The amount of people that you can treat due to the cost of the new techniques is very high. Some people are going one step back in technology to be able to make available for people treatment of cancer and others like vascular and anything because techniques are getting too expensive. So we have to provide techniques within the economic reality of countries. Doesn't matter the envelope so big, we have to make it available. And I think PDT, which is a well-known mechanism, can be one of those things to be available. Well, thank you very much, and I'm available for questions.